So hi, my name is Caitlin Willarth, and this is my poster on the morphological variation associated with pelvic structure polymorphism. My research is attempting to determine if the loss of pelvic spines is causing other morphological changes in the brook stickleback fish. So I have been working with John Mead to look into a question of biodiversity and ecology to see what is causing variation within a species. Variation is found everywhere throughout the world, no matter where you look. It's both within and between species and is really important for a species' persistence and its adaptive potential. So the differences in phenotypes are often determined by genetic factors, as an adaptive evolved response or as a plastic response to the current environment. Variation in body shape can reflect important ecological and behavioral differences among individuals, and this can have effects on their fitness. So the species I'm studying is the brook stickleback fish. It's one of 3 fish in its family that is also experiencing pelvic variation. Ancestrally, they all had pelvic structures, but some populations have evolved the complete loss of these pelvic spines. If you look at the photo in my background section on the left here, we can see 3 fish diagrams that all have varying amounts of red in them. These are ventral views, so we're looking at their bellies, and the red indicates the bony pelvic structure. We can see that there are 3 phenotypes here. On the left, we see a fish with a complete pelvic structure. We can see the girdle and 2 spines, one on either side of the body. And on the very right, we see no aspects of the pelvic structure. These pelvises are used for balance and maneuvering, but are mainly used for defending against predators. My hypothesis was that the presence of a complete pelvic structure would be associated with a morphological change, such as different body size or different body shape, compared to stickleback that were pelvic absent. The research is important as we want to know what is causing this variation. Are there certain populations or habitats that are so different that they're causing these changes? Or could the gene that is causing variation in pelvic structures have other pleiotropic effects? This question is why we expected to find a morphological response to pelvic variation. So to analyze the morphological variation, we collected 105 brook stickleback samples from 2 populations, Muir and Shunda Lake, Alberta. The samples were bleached and dyed with alizarin red, so the bones appeared red and they were clearly visible. The left lateral side of each fish was photographed in standardized conditions. And then 27 morphological landmarks were chosen, and all landmarks were digitally placed on each photo. The landmarks indicated an XY coordinate on a grid which would be used to compare the shapes of each fish to each other. So the morphometric analysis was performed using the geomorph package in the program R. The Procrustes and Nova assessment used a linear model to quantify the morphological variation that could be attributed to either one of 2 factors. So one, lake, so Muir or Shunda, and the second one is pelvic morphology, so present, absent, or vestigial. Our results were completely opposite from our expectations, as it turns out that the varying phenotypes have no association to a variation in morphology at all, although there were significant differences in the morphologies between the lakes. In my results section, the photo on the left shows the effect of pelvic spine on morphology. The landmark coordinates for all the samples with pelvic spines were originally placed on a symmetrical grid. Then the coordinates for the samples without spines were placed and plotted on top, which would have warped the results if there was significant shape difference between spined and unspined individuals. We can see that for the most part, the grids are mostly still perfect squares, and there's no statistical difference due to the pelvic phenotype. If we look at the graph to the right of that one, it shows the effect of lake. So the Muir Lake data was plotted onto the grid, Shunda Lake data was superimposed on top, and it warped the grid into the distorted shape we see here. This reflects the significant morphological differences of the fish between the 2 lakes. So while the pelvic spined fish were very similar to unspined, the shape of the fish from Muir Lake were quite different from the shape of the fish from Shunda Lake. The significant morphological differences between Muir Lake and Shunda Lake were apparent, but the cause still remains unknown. Since there's no distinct different habitats or feeding patterns for the stickleback with and without spines, it makes sense that pelvic spine polymorphism isn't associated with morphology. And this polymorphism is mainly maintained by predation pressures. While the 2 lakes are both north of Calgary, they're different in that Shunda Lake is a rocky mountain characterized with deep waters, and Muir Lake is more of like a shallow prairie lake. So variation between these lakes could either be an adaptive or a plastic response. The future direction of this research is really needed to determine if these 2 lakes are representative of all brook stickleback populations. But overall, the results of my study did not support my hypothesis and proved that further studies would really be needed to fully understand the situation of the diversity within these brook stickleback populations.